Hello everyone, Assalamu alaikum. In this video lecture, I am going to discuss with you about the different factors that are affecting the drug metabolism process. Factors influencing or affecting the drug metabolism. There are the different conditions or factors that will influence the metabolism of different drug molecules. Most important among them are the physicochemical properties of the drug, biochemical factors. Biochemical factors can be the enzyme induction and inhibition. And beside this, there are some other factors that are the biological factors, genetic variations and others. So now let's discuss them one by one. Number one, physicochemical properties of the drug molecule. The physicochemical properties of drug molecule, like its molecular size, molecular shape, lipophilicity, acidity, basicity value, its ionization constant and electronic characteristic, all of these properties have the influence on the metabolism of drug. As we have discussed the detail in our SAR lecture that how the size will affect the drug metabolism, because for example if the drug size is more larger, if drug molecular size more larger, then it may have the problem to bind with the specific site to its target, and therefore it cannot produce its effect. Same will be the case of the metabolism. It will not be properly metabolized, and that's why it can be accumulated and drug toxicity can occur. Besides this, there are the other properties that will influence the interaction of drug with the metabolism enzyme. Because of their size, lipophilicity, if the drug is highly lipophilic, it may causes the delay in the rate of metabolism, and that's why the duration of action of drug will be affected. Number two are the biochemical factors. Among the biochemical factors, the most important are the metabolic enzyme induction and metabolic enzyme inhibition. Metabolic enzyme induction is simply defined as any process that can increases the rate of metabolism of drug. It is known as metabolism enzyme induction. How the rate of metabolic enzyme induction has been induced? Because of increase in activity of metabolic enzyme, because of their activation or because of their stimulation. And due to the high rate of drug metabolism, ultimately the duration of action of the drug and its intensity will be reduced. There are the different type of drug or different type of agents that can be chemical or environmental that can access the enzyme inducers, means they are stimulating the metabolic enzyme to enhance their function. For example, among the drug molecule like barbiturate, the CNS stimulant, that is the alcohol molecule, pentobarbital, phenytoin, cortisol, testosterone, coumarin, all of these have the ability to stimulate the drug metabolism enzyme. And due to the activation of some specific enzyme by these specific drugs, ultimately the rate of metabolism of those drugs will be enhanced, which are being metabolized by that specific enzyme. There are certain environmental factors such as pesticide, DDT and polycyclic aromatic hydrocarbon that are present in the cigarette smoker. They also act as the enzyme inducer, means the pesticide DDT and if someone is smoking cigarette then and taking certain drugs, then there are the chances of high rate of drug metabolism if they are taking certain drugs. Metabolic enzyme inhibition, this is opposite to the metabolic enzyme induction. There are the certain type of drugs that and environmental factors that can cause the metabolic enzyme inhibition, means now the activity of metabolic enzyme is being reduced, being decreased, and due to their decrease in activity, ultimately the metabolic rate of the drug will also be reduced, decreased or diminished. So that's why the drug may get accumulate inside the body, or it will slowly metabolize. Means now the duration of action of the drug will get prolonged. For example, the methacholine, that is one of the antihistaminic drug, it inhibits the metabolism of acetylcholine. Why? Because they both are competing for the enzyme cholinesterase. Similarly, the isoniazid, that is the anti-TB drug, inhibit the metabolism of phenytoin. So means if these both drugs are given parallel, then isoniazid will ultimately inhibit the metabolism of phenytoin because of competing with the same enzyme for their metabolism. Such influence of one drug on the metabolism of other drug may lead to drug-drug interactions. Among the environmental factors like heavy metals, nickel, mercury, arsenic, they also have the tendency to cause the inhibition of certain metabolic enzyme, and ultimately they will reduce the metabolism rate of those drugs that are being metabolized by that specific enzyme. Biological factor. Among the biological factors that are influencing the metabolism rate are the different species differences, strain differences, pharmacogenetics, diet, pregnancy, hormonal imbalance, presence of disease state and or not. So these are the different factors. For example, during the pregnancy there are the hormonal changes occur inside the woman body. So due to these hormonal changes, ultimately they will affect the activity of the drug metabolism. Again, if someone is healthy, their metabolism rate will be higher in comparison to those who are suffering from certain diseases. For example, let's consider the liver patient. The liver patient or the kidney patient or the other chronic disease patient will have the problem in the metabolism rate of the drug because of the decrease in activity of the specific metabolic enzyme. And the other example for the effect of pregnancy on the drug metabolism is during the third trimester of the pregnancy, the anticonvulsant drug carbamazepine, phenytoin, their metabolism rate is being affected due to the hormonal changes inside the human body. Now let's discuss the important consequences of the drug metabolism. Means what are the end product are the for the after the drug metabolism, means how the drug can be metabolized, and after metabolism what can be their end product. And among the important consequences, one of it is the pharmacological inactivation of the drug. Majorly after the drug metabolism, the drug is get converted into its inactive form, that is readily excreted out. The metabolite that form has maybe the little or absolutely no pharmacological activity. For example, the phenytoin, that is one of the anti-epileptic drug, it get converted into para-hydroxyphenytoin, which have no any pharmacological activity. The other consequences is no effect on pharmacological activity. Here the metabolite form after the metabolism reaction may have the equal activity or similar activity to that of original drug. Means in this condition there is no any change in the pharmacological activity, no any decrease or no any increase in pharmacological activity. For example, the nortriptyline that is a drug that contain the equal activity similar to its parent drug, that is the amitriptyline. So basically amitriptyline is one of the antidepressant drug. When it get metabolized, it is converted it into its metabolite, that is the nortriptyline, and its activity, that is pharmacological activity, is similar to the parent drug. Toxicological activation of drug. This is one of the most important factors that sometime after the metabolism, the drug metabolite that are formed have the higher tissue reactivity. Due to the higher tissue reactivity, they may cause the toxicity. For example, the toxicity reaction caused by the toxicological activation of the drug can be the necrosis, can be the hepatitis, that is means the prolonged exposure to the liver tissues, a certain type of reaction, and ultimately they may causes the liver tissue, the hepatocytes damage, and ultimately they will lead to the hepatitis, that is the inflammation of the liver cell. For example, the N-acetine parabenzoquinone, it is the one of the metabolite of the paracetamol that may lead to the hepatotoxicity. In SAR lecture, we have discussed that how can we overcome the paracetamol hepatotoxicity. Pharmacological activation of drug is basically this refer to the special type of drug molecule, that is the product that itself are inactive but they converted into the more active form that is pharmacological active inside the human body, inside the in vivo. Where you, for the detail of the prodrug, you may refer to the link given here. For in the other lecture we have discussed their types and why we are going to synthesize or design the product molecule. The factors are to overcome their unattractive physicochemical properties, to overcome their pharmacological ADR, and to attain the optimal bioavailability. For example, the ampicillin, that is one of the antibiotic, it is the active form of ampicillin. The next consequences can be the change pharmacological activity. In this condition, after the metabolism, the metabolite that have completely different pharmacological activity than the original drug. For example, isoniazid, that is one of the anti-TB drug, is converted into one of the metabolite, that is the aprionazid. Aprionazid, on the other hand, has the antidepressant pharmacological activity that is totally different from the anti-TB, that is the one of antimicrobial activity. Now let's have a quiz. So these are some basic questions from this lecture. You have to attempt these questions in the comment section, and besides this, if you have any question, you may ask in the comment section. Question number 1: name the different factors that are affecting the drug metabolism. Question number 2: give the example of metabolic enzyme inducers and metabolic enzyme inhibitors. Question number 3: which environmental chemical can cause or can act as metabolic enzyme inducer and enzyme inhibitor. Question number 4: define toxicological activation of drug.